Welcome back to Cancer as a Mitochondrial Metabolic Disease Basics. Ivermectin is usually discussed in one of two ways, either as a suppressed miracle or as a dog dewormer and misinformation. But I think both sides are asking the wrong question. What caught my attention was not the above back and forth. It was a cancer study showing that ivermectin reduced the expression of multiple key enzymes involved in the TCA cycle and a little known energy pathway called mitochondrial substrate-level phosphorylation. That is where glutamine is fermented. And that immediately made me wonder, could ivermectin be interfering with one of cancer's main energy producing systems? Let me get this one thing out of the way first. Ivermectin was approved by the US Food and Drug Administration in 1987 for humans against river blindness. And today, it remains FDA approved for the treatment of several human parasitic infections. But that's not what today's discussion is about. Today's question is biochemical. Can ivermectin disrupt cancer's mitochondrial energy network? Specifically, can it interfere with the systems that allow cancer cells to keep making ATP, or cellular energy, when oxygen is low and normal oxidative phosphorylation is compromised? So the real question is ivermectin just another repurposed drug with limited or no anti-cancer activity? Or could it be a critical component to a larger metabolic strategy to collapse cancer's hidden energy rewiring diagram? We will soon find out. So without further ado, let's get into it. So today we are going to uncover something that makes ivermectin very unique, and that is its ability to interfere with cancer's bioenergetic networks. And I'm going to explain what I mean by that. We have looked in prior videos about how ivermectin can affect multiple key signaling pathways. We have also looked in the past at how ivermectin can induce oxidative stress and activate apoptosis through the caspase dependent systems. Here's another graphic looking at that. We have also looked in the past about how ivermectin can affect the glycolysis system, or the fermentation of glucose within a cancer context. But it wasn't until a recent series of papers that I started reading. This is one of them. I posted the other ones on the community post that really challenged me to look at how this system functionally works. And the system I'm referring to is called mitochondrial substrate-level phosphorylation, or the fermentation of glutamine. And I will say that many people are aware of glycolysis because it's probably one of the most basic biochemical metabolism networks talked about in many levels of education, whether it be high school, college, and beyond. But glutaminolysis, or the breakdown of glutamine, is much more specialized and not widely taught or well understood. So I'm going to quickly break down what is at play here. So glutamine is the most abundant amino acid in the bloodstream. And the glutamine from the bloodstream has to get into the cells through a glutamine or an amino acid transporter. And that's what these represent: SLC1A5, 3A1, 6A14. These are glutamine transporters, or amino acid transporters. And we have looked in the past about several things that have had a down-regulatory effect on, let's say, SLC1A5 or SLC38A1, etc. And we have also then looked at how once glutamine is inside the cell, has to be converted into glutamate via an enzyme called glutaminase, or GLS. And this is where DAWN specifically blocks, as well as other things, have effects here. What we have not talked as much about is what happens once glutamate is created. There are multiple fates for glutamate. It's a very critical amino acid, but the fate that we're going to be talking about is what happens when glutamine through glutamate is fermented to make ATP through mitochondrial substrate level phosphorylation. Well, that takes a couple additional steps. We have to go from glutamate to alpha-ketoglutarate, alpha-ketoglutarate to succinyl-CoA, and then succinyl-CoA to succinate. And that's not very well shown in this graphic, but the bottom line is there's a series of steps that get us through that to happen. And what's also less well known, and what some of these more recent papers that I'm referring to have helped me better understand, is that although oxidative phosphorylation, or the making of energy using oxygen through an intact functional mitochondria with good cristae and supercomplex formation, even though that system is compromised and ATP is not being made through that system at any appreciable rate, what is happening is that parts of the electron transport chain is still operational. Specifically, in this particular talk, I'm going to talk about complex 1. And what this paper made me realize is that there are more targets for us to look at to help shut this system down. So here is the same graphic as this, but it's kind of inverted. So glutamine uptake is over here, glutaminase is here, glutamine dehydrogenase is here. Here's alpha-ketoglutarate, and then it has to go through alpha-ketoglutarate dehydrogenase complex to get succinyl-CoA. Succinyl-CoA has to be converted through succinyl-CoA ligase to succinate. Here is where ATP is made. This arrow is being pointed at. This is where ATP is being made. And this is where NAD must be used in this system. And it begs the question, and this is what these biochemical researchers are actually asking, is where is NAD recycled if the electron transport chain is shut down? And basically what they found is that the electron transport chain, pieces of it, still can work without oxygen. And specifically complex 1 can change NADH to NAD to allow NAD to be freed up so it can work with this system. So this is now the second or third time we've inverted this, but this is the electron transport chain. We have no oxygen or limited function here. But complex 1 is still functioning to recycle NAD to allow for alpha-ketoglutarate to be converted to succinyl-CoA and succinyl-CoA to be converted to succinate to make ATP. This is the mitochondrial substrate-level phosphorylation of glutamine. But in order for the system to work, it requires NAD. And that is where complex 1 comes into play. It also requires this enzyme here, this KGDHC, this alpha-ketoglutarate dehydrogenase complex, as well as SUCL, which is succinyl-CoA ligase. And that's where it gets very interesting for ivermectin specifically. This paper is a paper that I actually did present on about a year ago, but I don't think I truly appreciated what was actually being shown here. So it's titled Pharmacoproteomics Reveals Energy Metabolism Pathways as Therapeutic Targets of Ivermectin in Ovarian Cancer Towards 3P Medical Approaches. And it says here in the abstract, integrating our previous clinical ovarian cancer tissue and cell experimental data, this study found that ivermectin significantly interfered with the energy metabolic pathways of ovarian cancer, including glycolysis, TCA cycle, oxidative phosphorylation, and other related pathways. And this paper was pretty thorough. It had all the different metabolic networks, and it showed exactly where ivermectin was having an effect and to what degree it was having an effect at the doses used in these mouse studies. And what I'm particularly interested in for this talk is what's circled here in red. This is where glutamine convert to glutamate, glutamate into alpha-ketoglutarate, which has to be then converted by these enzymes into succinate to make ATP. These are the enzyme systems are key to mitochondrial substrate-level phosphorylation, and that's exactly where ivermectin has some of its largest effects. In fact, it had a table in this paper of all the proteins. I've only clipped out the TCA cycle proteins, and I circled in red the exact subunits of the proteins that are responsible for those key enzymes that are participating in mitochondrial substrate-level phosphorylation. And then what I did was I looked at what happened in the treatment group versus the control group and what the expression differences was. And I actually had a table be put together based on that information. So for alpha ketoglutarate dehydrogenase complex, okay, let's just get back into this paper here. This is the enzyme complex we're talking about. Glutamine to glutamate, glutamate to alpha ketoglutarate. Alpha ketoglutarate has to be converted to succinyl-CoA as part of this mitochondrial substrate level phosphorylation. This is the enzyme we're talking about. With ivermectin added, it's lowered the protein expression of the enzyme by 50 to 60%. And the next reaction, which is the succinyl-CoA to succinate reaction through this SUCL or succinyl-CoA ligase reaction, this reaction here, which is critical to make ATP from glutamine, is lowered from 60 to 70% with the doses used in the study. It then looked at, what they actually did. They gave ivermectin, they looked at the body weight, they looked at the cells. I'm sorry about this, the graphics are so bad because this paper, unfortunately, was behind a paywall. So it only gave the graphics that were available for free on the PubMed website. So when you blow them up, they look terrible. But basically what they did was they gave them nothing. They gave them either 5 or 10 milligrams per kilogram of ivermectin. So the light blue line would be this 5 milligrams per kilogram, and this would be 10 milligrams per kilogram, the dark blue line. And you see that the tumor size, if you let it go after 15 days, was about, 900, 850. Again, terrible graphic. And at 5 milligrams per kilogram, there was about a 50% decrease in tumor size. And at 10 milligrams per kilogram in this study, it was about a 70% decrease in tumor size at 15 days in this mouse study at these respective doses. Well, then what I did was I used this body surface area allometric conversion. That is an FDA conversion from mice to human studies to give you a ballpark equivalent dose of what 10 milligrams per kilogram in a mouse would be for a human. And it ended up being 0.81 milligrams per kilogram. Now, the reason I chuckle is because the proposed dose by some of these researchers, for example, this is one of my favorite papers that was published in the Journal of Orthomolecular Medicine called Targeting the Mitochondrial Stem Cell Connection in Cancer Treatment, a Hybrid Orthomolecular Protocol. And I went ahead and clipped out the part where they are proposing to use the off-label drugs for cancer. And what they were saying was that ivermectin is a very safe drug. In healthy volunteers, a single dose was increased to 2 milligrams per kilogram, and no serious reactions were found. Demonstrated in another study, cancer patients who took ivermectin at 5 times the standard dose, up to 1 milligrams per kilogram daily for up to 180 consecutive days, had no serious side effects. My point is that other literature will say that the proposed anti-cancer dose of ivermectin is somewhere as low as 0.3 milligrams per kilogram and maybe as high as 2 milligrams per kilogram. Now, I'm not saying that you should use any of this. I'm not saying that there cannot be side effects with even the lower doses of it. I've seen it. The bottom line is that the estimated human equivalent dose of ivermectin that best correlates with the 10 milligrams per kilogram, which was about a 70% decrease in tumor size at 15 days for these mice, was 0.81 milligrams per kilogram, which is right in the middle of the proposed therapeutic dose of ivermectin by several research groups, including this paper here. I do think one of the final frontiers for ketogenic metabolic or press pulse metabolic therapy is on the glutamine side. DAWN exists. It is a sledgehammer. It's going to affect all of glutaminolysis. That is good and bad. We have EGCG and other things that affect glutamine dehydrogenase. We do not have tools for this GOT2 or GPT2. No effective tools for that. But if we get into the nitty-gritty of really what matters for mitochondrial substrate level phosphorylation or the fermentation of glutamine, these key TCA cycle pathways are really the deepest portion of the system. And ivermectin seems to have a significant effect at the level of 50 to 60% decrease expression at therapeutic doses of potentially lowering these enzymes, which I think, again, does not mean that it by itself is going to be some miracle. I don't believe that's the case. But I do think that it could be an important component to a therapeutic strategy, also known as metabolic therapy for cancer. That I do believe is possible. Only time and study will tell. If you like videos like this, please like, share, and subscribe. And until next time.